model. This is the interior of one skeletal muscle cell also. Wow, I was hoping I'd be over this by now. Last Thursday night I couldn't talk at all. The laryngitis was so bad. So you probably think, oh damn, it didn't last until this Thursday. I got to hear this. So, all right, so here we are looking at the interior of one skeletal muscle cell. So that's sarcolemma. This is cell membrane. This is endomysium connective tissue wrapping around the cell membrane right here. Take this out. Come on, it should slide out. And rippled portion of the sarcolemma, that's the motor end plate right here, where all of the nicotinic cholinergic receptors are located. So every little dot you see represents one nicotinic cholinergic receptor, a ligand-gated sodium ion channel right there. So again, motor end plate portion of the sarcolemma, and then the rest of the sarcolemma is where the voltage-gated ion channels will be for the generation of the action potential. Okay, and guess what these are? Guess what this is? This. Myofibrils, okay? They're log-like objects that run the length of the cell. So cross-sectional view through 1, 2, 3, 4, 5 myofibrils here. And you can see there are mitochondria wedged in there, and the gray stuff, the blue-gray stuff, that's what we now call the sarcoplasmic reticulum. More on that shortly. But this cross-sectional view reveals the larger orange dots and the smaller blue-gray dots. Larger orange dots are cross-section through thick myofilaments. Smaller blue-gray dots, cross-sectional view through thin myofilaments, composing one myofibril here. So obviously, they clearly overlap here. Do they overlap continuously through the length of the myofibril? No, they don't. So they just happen to cut through the cell where the thick and thin myofilaments were overlapping one another. They don't always do that. So in other words, you can cut along some other points on this model and only get thin or only get thick myofilaments in cross-sectional view. For example, I'll get this off right now. For example, this view right here. There's an I band. There's another I band. So this is a Z disc. Look, we've got the Z disc going continuously through myofibril to myofibril to myofibril. One continuous large protein sheet. Same thing with the Z disc over here. One large continuous protein sheet. So Z disc in the center of these I bands. One Z disc in the center of these I bands. Here's an A band right there. Here's an A band with another myofibril. And so there's the M line, a much smaller protein sheet that only holds its own A band together. So this M line is unique to this A band, and so on. So an A band is a bundle of thick myofilaments. So this thing right here, this pale orange band, this pale orange band, this recessed pale orange, actually it's a darker orange band, I beg your pardon. This orange band right there, that's one thick myofilament. There's another thick myofilament. There's another thick myofilament. Okay, from there to there. So thick myofilaments run the width of an A band. Thin myofilaments connect to a Z disc. So there's a thin myofilament in gray coming out from this Z disc, and there's a partner going the opposite direction from the other side of the Z disc. So you can see thin myofilaments projecting from both sides of a Z disc. Here, if we were to cut through here, all we would get in cross-section would be thin myofilaments. If we were to cut through here, all we would get would be thick myofilaments. But this view is obviously cut through a place like there, where they're overlapping one another. You see thick out to there, thin into there. So they're clearly overlapping one another at that point. So even in a relaxed state, there's partial overlap between thick and thin myofilaments. That's necessary for contraction because they have to link to each other. If they don't have at some point where they're overlapping, they can't connect to one another. Let's see, so sarcoplasmic reticulum shown here. More of sarcoplasmic reticulum, all the blue-gray. You guys learned about, in Bio 110, you learned about the smooth endoplasmic reticulum. In most cells, it's the source of membrane lipids. In muscle cells, the smooth endoplasmic reticulum has a totally different function, so we give it a totally different name, sarcoplasmic reticulum. It stores calcium ions. It uses calcium ATP as its primary active transport to store calcium ions. So lots of calcium ions are packed within the sarcoplasmic reticulum. And let's see these holes in the sarcolemma? Hole there, hole here, here, and so on. So that hole represents the sarcolemma actually folding inward, creating a membrane tube. This is a continuation of the sarcolemma. There's another continuation of the sarcolemma internally, a third one, a fourth one. Each of these is called a transverse tubule, running transversely, perpendicularly to the long axis of the cell. So usually just contracted from the T tubule, T tubule, T tubule, T tubule. You can see each T tubule has sarcoplasmic reticulum bulging against it. Sarcoplasmic reticulum here on either side of the T tubule. So a bulge right there on sarcoplasmic reticulum is called a terminal cisterna. Here's another terminal cisterna. And in between them, T tubule. Cisterna, tubule, cisterna. Cisterna, tubule, cisterna. Cisterna, tubule, cisterna. So that triple structure is actually called a triad. So we're showing 1, 2, 3, 4 triads. Well, there are many more. If we looked at we backed up and saw the entire cell. So again, a T tubule is a continuation of the sarcolemma internally, and it butts up against a terminal cisterna of the sarcoplasmic reticulum on either side of it. And the majority of the calcium ions are in these terminal cisternae. So when we have an axon terminal of an axon collateral of a somatic motor neuron right here, and all the yellow dots in here are synaptic vesicles filled with acetylcholine. That, by the way, is the nucleus of a Schwann cell. Schwann cells actually do attach to axon terminals, but they never myelinate them. So that is not part of the neuron. This is actually part of the Schwann cell right there. So you've got the axon, there's another Schwann cell forming the myelin sheath, teledendron to axon terminal, mitochondria, of course. So an axon potential gets to here, gets to the axon terminal, and that movement of calcium ions in, exocytosis of ACH all over the motor endplate, binding to all of these little teeny tiny nicotinic cholinergic receptors. Depolarizing local potential spreads out, gets to the sarcolemma beyond motor endplate where the voltage gated channels are. They open, bang, action potential spreading out, and guess what? Action potential continues down inside the cell along the T tubules, opening up voltage gated channels in the terminal cisternae, and out comes gushing calcium ions into the cytosol of the muscle cell. So calcium is critical for contraction. The action potential releases the calcium from the sarcoplasmic reticulum. Okay, and then what actually happens is this. The calcium is necessary to allow the overlapping thin and thick myofilament to join, to bind, to form linkages. And so when they link, they start to slide together. The thin filament here slides toward the M line. Thin filament here slides toward the M line. So what happens to the H-zone? During contraction of the sarcomere, Z to Z, the myofibril, during contraction of the sarcomere, the thin filaments are moving in toward the M line, and the sarcomere is contracting. But what is happening to the width of the H-zone?
It disappears and becomes dark.
It's getting noticeably narrower. So as the Z discs are being pulled in, as the thin filaments ride the thick filaments toward the center, the H-zone gets narrower and narrower and narrower. A fully contracted sarcomere has no H-zone. The thin filaments go all the way in to the M line. No more H-zone when the sarcomere finishes contracting. Okay, so what this shows you is a myofibril, a Z disc, thin myofilament, thin myofilament, thin myofilament, thin myofilament. Here we've got an A band, actually, I beg your pardon. Thick myofilament, thick myofilament, thick myofilament. M line, H zone, right there, where there's no overlap between thick and thin. And here we do have overlap between thick and thin. So when thin and thick myofilament link, they form what are called cross bridges. The thin filaments begin to slide in toward the M line like so. Look what's happening to the H zone. It's getting narrower, getting narrower, getting narrower. Now, this won't let me push it in anymore, so it stops too soon on this model. The thin filament should go all the way in to the M line. There should be no more H zone, period, upon full contraction of the sarcomere. So yes, I put a lot of nits on these models because they're worth pointing out. Questions?
What was the triad again?
Pardon?
The triad, what was it again?
The triad is a combination of a terminal cisterna, a T-tubule, and a terminal cisterna. So, that's up, is that over here? Yeah, it's over here, it's on this side. So, terminal cisterna, T-tubule in the middle, terminal cisterna. That's one triad. Anything else? Let's grab